Okay, hello everyone. So this is the cancer genetics lecture from the human genetics module, which is a level 1 module at the University of Bradford on the biomedical sciences degree programme. So the aim of this lecture is to give you a basic introduction that should enable you to understand a little bit more about how genetics contributes to the causes of cancer, to the etiology of cancer as a disease. And what you should be able to do at the end of the lecture is discuss at a basic level how cancer cells arise, and in particular be able to discuss and talk about how genetic factors contribute to the formation of cancer and impinge and alter cell growth in general, leading to the development of tumours and cancer as a disease. So you should all be aware, I imagine you're all aware, how devastating cancer can be a disease. This is an individual unfortunate enough to have testicular cancer. But one of the things that we're going to consider in the next lecture on gene therapy is how our understanding of molecular biology and genetics is enabling us to fight back against this disease. And the good news about cancer is that we are improving all the time survival rates for individuals who develop different types of cancer. This individual, if you've not recognised him already, is Lance Armstrong. So he's a famous cancer survivor who went on to win 6 Tour de France titles. Here he's celebrating one of them with a glass of champagne. However, if you read the news, obviously the champagne he's drinking here may have contained other elements as well, allegedly. And just goes to show you that even cancer survivors, who can often seem very heroic, are still human. So let's start then with just a brief overview of what we're going to talk about in the lecture. We're going to start by discussing how a cancer cell becomes cancerous in the first place. And in particular, we're going to have a look at what's going on at the molecular level of the cell. So what molecular changes are occurring that are causing that cell to start dividing in an uncontrolled manner and forming a tumour. So briefly, the genetics of cancer as we're going to consider them today are going to include a discussion and an explanation of what oncogenes are, a discussion and an explanation of what tumour suppressor genes are, and how importantly these two different factors kind of work almost in opposition to one another in order to regulate cell growth. We're going to consider also the multi-step model of cancer as a disease. So this is a concept that a single mutation is enabled to cause cancer as a disease in ourselves. We actually require numerous mutations in order to develop an invasive cancer that's going to kill us. And we'll finish by summarising what we've talked about in the lecture. So how do cancer cells arise then? Well, we've had an entire lecture on the cell cycle, and I've mentioned at numerous points throughout this module how tightly regulated cell growth is at the molecular level by the cell. So you should be aware now that the cell cycle and its numerous checkpoint systems does a pretty good job of making sure that cell division only occurs in response to the correct signals, and when it does occur, it occurs without error in the correct order. So normally cell growth is a very regulated process, but occasionally this process can be derailed. That might be due to external factors such as mutagens, radiation affecting the integrity of the DNA. It could be due to inherited mutations in the DNA which affects the cell's ability to monitor and control cell growth properly. But for any number of reasons, occasionally cell growth is derailed and cell division and cell growth starts occurring in an uncontrolled manner. This is what we understand as cancer. So to give you a couple of terms that you should become, if you're not already become familiar with, the cell growth of this type, new cell, uncontrolled cell growth, is often referred to as a formation of tumours or neoplasms, which effectively means new growth. Now it's interesting to think about cancer actually as cellular microevolution. So if you think about what we consider to be natural selection, evolution, the selection of the fittest over the less fit, and think about what's going on at the cellular level with cancer, there are a lot of parallels there. So if the aim is in life to develop an organism that's faster and quicker than the next organism in line, and that the faster and quicker organism, the stronger organism is going to be selected out, and that's the one that's going to survive. A similar situation is occurring at the cellular level with cancer. So if we have cells which are all growing in a tissue, and one of those cells gains the ability suddenly to divide much quicker and much more voraciously than its neighbours, that cell is going to take over that tissue expanse. And that's effectively what's happening when a tumour forms. So there's a strong underpinning natural selection actually for the formation of tumours. So you could argue that all human beings are effectively just tumours waiting to happen. Now, in the opposition to that, obviously, there wouldn't be much point having an organism that just, as soon as it was born, developed tumours and died. We need a way of keeping those tumours in check, controlling that desire, if you like, for the cells to evolve and just divide in an uncontrolled fashion. And that's how over millions of years we've evolved lots of different complex mechanisms for making sure that cancerous cells, tumour cells, are either destroyed or prevented from forming in the first place. Certainly until we've gone past a reproductive age. As we get into old age, where most cancers affect us, the need for natural selection is diminished somewhat. We're no longer at an age where we're going to reproduce. So it's no coincidence that cancer is considered a disease of the elderly, primarily, and that you don't normally see tumours forming until late in life, after the reproductive age of the actual individual has long since passed. So another word that you need to be aware of, another term, is transformation. So the process which leads to a cell ceasing to be normal, if you like, inverted commas, and becoming cancerous is termed transformation. So if a cell is said to have undergone transformation, that means that the cell has gone from a normal state to a cancerous state. It's gone into a state where it's dividing in an uncontrolled manner. Now, it's important that you don't confuse this term with the identical term transformation, which you may encounter in bacterial lectures. So the process of introducing DNA into bacterial cells, exogenous DNA, artificial DNA if you like, into bacterial cells is also called transformation. That's a different transformation to what we're talking about here. With regards to cancer biology, when you talk about cellular transformation, you're talking about the transformation of a cell from a normal state to a cancerous one. And you're probably also aware that cancers can be described as benign or metastatic. You may not be aware formally of what the definition of those two terms are. A benign cancer is effectively restricted to a clump of cells. So this is where a cell has gained the ability to divide in an uncontrolled manner, and it divides in that way very rapidly to form a tumour in the body, in the tissue, in the brain, in the lung, in the kidney, wherever it happens to arise. But importantly, this tumour that forms, this clump of cells, remains as a single entity in the body. It doesn't go off elsewhere in the body and cause additional tumours to form. So for that reason, benign tumours are generally relatively safe. You wouldn't want one, you wouldn't wish one on yourself, obviously, but generally benign tumours aren't going to kill you, for the simple reason that surgery can often remove the tumour in its entirety, maybe some follow-up from chemotherapy to get rid of any residual tumour that's been left over by the surgeon, and the patient won't go on then to relapse and develop further cancers in the future. Metastatic disease is a much bigger problem. So metastatic cancers, as we'll see towards the end of the lecture, are cancer cells which have gained the ability to break off from this initial clump of cells which forms in a benign tumour and drift around the body in the bloodstream or the lymphatic system, establishing new tumours at distal sites from where the original tumour formed. So when you hear of tumours and cancers spreading around the body, these cancers are cancer cells which have become metastatic, gain the ability to break off from the first tumour which has formed and spread to other locations around the individual. And these cancers are much more difficult to treat because it's impossible often to remove them completely by surgery, and even a small amount of cancer that's left over following chemotherapy or radiotherapy can establish a new tumour and then form new metastatic disease again, which is why patients often relapse if they're suffering from metastatic disease. So as a general rule of thumb, benign cancer good, metastatic cancer bad, but it's, as everything in biology, there's always exceptions to the rule. So for example, there are situations where benign tumours can form in tissues which we can't operate on easily, the most obvious example being brain tumours. And if you get unfortunate enough again to develop a brain tumour that's benign, even though that tumour is not going to metastasize and spread to other locations in the body, it may still kill you for the simple reason that we're unable to operate on this tumour to remove it. And if the tumour continues to grow, expand into the brain matter to a significant level, it's likely to seriously affect the health of this individual and may ultimately result in their death. So benign tumours, even though inverted commas they're relatively safe a lot of the time, if they appear in locations which impinge on essential cells or in areas which are difficult to operate on, can themselves be deadly to the individual. So a few terms there for you: neoplasms, transformation, benign and metastatic disease that you need to be very familiar with as basic terms when we're talking about cancer. So obviously this disease wouldn't be on this module if it didn't have a genetic basis. You could argue that cancer is almost the most important genetic disease of them all. It's certainly going to affect most people during their lives in some way, whether it's personally or via a relative. 1 in 3 people who are listening to this podcast will die of cancer. That's a proven statistic. So it's a disease which affects everybody in the global population, and importantly, it's a disease of genetics. So how do we know that as a fact? Well, there are several observations that have been made over the years with regards to cancer, which inform us that it's definitely a genetic disease. So one of the first things that people realised was that certain types of cancer, not all cancer, but certain types of cancer certain types of cancer have a predictable pattern of inheritance in family trees. So you can track the appearance of cancer through family trees in the same way that you track a known genetic disorder such as cystic fibrosis or phenylketonuria. Certain cancers can be tracked in the same way through family trees. And it turns out that these types of cancers are these familial cancers. And the familial cancers actually only form a very small part of all cancers which affect people in the global population. Familial cancers, compared to the sporadic cancers, which is the other type of cancers, are very rare. Far more likely that you will develop a cancer sporadically than you will inherit it from a parent or a grandparent. But these familial cancers do exist. We'll give some examples later on. And when we look at their inheritance pattern through family trees, they're similar to the inheritance pattern of known genetic disorders. So that implies that some cancers have a genetic mode of inheritance. A really important observation that led people to make the leap, if you like, or to confirm people's suspicions that cancer was a genetic disease, is the fact that if you infect individuals with certain types of viruses called oncoviruses, then those individuals go on to develop cancer. So the most famous example of this is, or if you like, the first example of this was the identification of a viral gene in the virus Rous sarcoma virus that, when expressed in infected individuals, led to the formation of a sarcoma, which is a particular type of bone cancer. So if viruses were able to induce cancer by infecting individuals, that implied that the genes which the viruses expressed following infection were actually probably the agents that were causing the development of the disease, of the cancer, and therefore again supported this hypothesis that cancer has a genetic basis. It goes on, cancer cells are actually clonal. So once a cell becomes cancerous, when it divides and to develop 2 new cells, both of those cells are cancerous as well. So the thing that's making the cell cancerous is passed on during cell division, through generations. So therefore, the thing which is making the cell cancerous must be genetic in basis if it's been inherited by all subsequent daughter cells. So the fact that cancer cells are what are termed clonal is another big clue that cancer is caused by defects to the genetic material in the cell. This is supported by experiments which have shown that if you expose cells to chemicals which are known to damage DNA, so-called mutagens, then the cells which are exposed to these mutagens often go on to display cancerous-like phenotypes. The cells will become transformed, start dividing in an uncontrolled manner, and effectively develop into cancer. Obviously, the same is true if you're exposed an entire individual to mutagens, be that radiation due to extreme examples like Chernobyl, or smaller examples such as what happened to Marie Curie during her work with radiation in the 19th century and turn of the 20th century. These mutagens eventually will cause enough DNA damage to accumulate, and the accumulation of the DNA damage will manifest ultimately as cancer in those individuals and kill the individual. So chemicals that damage DNA lead to cancer. More evidence that cancer is a genetic disease. And finally, we also know that if you start translocating sections of chromosomes around inside the nucleus, you can often cause cancer to develop as a disease. The classic example of this we talked about earlier in the module, and that is the translocation of the Philadelphia chromosome to generate the BCR-ABL fusion protein. So we talked about BCR-ABL when we talked about single gene disorders. And that's a classical example of how chromosomal rearrangements can lead to the development of cancer. In that instance, a chromosomal rearrangement places two genes next to one another to form what's called a chimeric gene, this BCR-ABL or oncoprotein. We'll talk about oncogenes later on in this lecture. In other cases, it might be that when a chromosomal translocation event places a gene which is normally switched off into a region of a chromosome which is extremely active for gene expression, has very loose chromatin structure, and this causes activation of this gene in an aberrant manner and subsequently leads to aberrant cell division and cancer. So we'll talk about how different activation and repression of genes can lead to cancer over the next few slides when we talk about oncogenes and tumour suppressor proteins. So what's going on at the molecular level then? So we've got lots of evidence on that last slide of why we consider cancer to be a genetic disorder. And we know that cancer cells are clonal. We know that when a cancer cell divides, it produces 2 more cancer cells, and they produce 4 more and 8 more. But what's going on at the molecular level to actually turn a cell from being normal into being cancerous? Well, effectively what's going on is the cell's accumulating mutations that are allowing it to effectively evade the checkpoint mechanisms of the cell cycle. So that's one of the major events which a cell must go past in order to become cancerous. It needs a way of evading these checkpoint mechanisms. And we've talked about these checkpoint mechanisms at length in the previous lecture on the cell cycle. They're relatively complex in nature, and what the checkpoints do is they act as a brake on the cell cycle machinery. So if this is the cell cycle machinery, if when cells divide they go round this machine, round through the cell cycle in a continual fashion, as long as growth factor stimulation is being provided, what the checkpoint controls do is they act as a brake to this cycle. They're able to stop the cell from dividing at specific points in response to certain stimuli or certain signals. And there are 3 major checkpoints which we discussed in the cell cycle lecture. The G1S phase checkpoint, the checkpoint that exists at G2M phase, and the so-called mitotic spindle checkpoint. And all 3 of these things are involved in making sure that if a cell is not ready to divide, it doesn't divide. And the reason that they're checking that that's the case is that they want to avoid the possibility of that cell becoming cancerous, of that cell gaining mutations which then enable it to just completely disengage from this process and just divide in a completely uncontrolled fashion. Now, we talked about metastatic and benign cancers and how they're different. It's often the case that the most, if you like, nasty type of cancer cells, the cancer cells which are very metastatic and very prone to spreading around the body of a patient who's unfortunate enough to have metastatic disease, are defective in all 3 of these checkpoint control mechanisms. So it's not just a case that one of these goes out of order. Often if one's taken out, another one can compensate to a certain extent. But the most aggressive types of cancers will often have acquired mutations which disable all 3 of these checkpoint control mechanisms. So what proteins then regulate these checkpoint control mechanisms? How are these themselves actually working? Well, the proteins responsible for actually monitoring the checkpoints within the cell cycle, or at least some of the proteins which are responsible for monitoring those checkpoints, are what we term tumour suppressor genes and tumour suppressor proteins. And we're going to discuss what they are in a couple of slides' time. So before we talk about tumour suppressors, which, as I've just said, act as a brake on the cell cycle, I want to first talk a little bit about oncogenes, which you may or may not have encountered during A-level, I'm not sure. So oncogenes are genes that trigger cellular transformation when they're expressed. So if you express an oncogene in a cell, it's got the ability to cause that cell to become cancerous. And they were originally identified by virologists who were studying a particular type of family of viruses called retroviruses. So the virus I mentioned earlier on in the lecture, this Rous sarcoma virus, this particular virus is a part of the retrovirus family. And it was work studying Roux-sarcoma virus that led scientists to realise that when the virus infected people, one of the genes that it was expressing was actually responsible for causing the cells it infected to become cancerous. And this particular viral gene was called SARC, SRC, all lowercase. And it turns out that what SARC is one of these so-called oncogenes. We now actually segregate oncogenes into two families. There are viral oncogenes and there are actually cellular oncogenes, which we'll talk about in a moment. And depending on which you are looking at in the textbooks, the viral oncogenes will be referred to as a V dash and then the name of the gene. So, for example, SARC from Roux-sarcoma virus is written V for virus hyphen SRC. Often now we realise that there are cellular equivalents of these oncogenes. If you're talking about a cellular oncogene, then it is often referred to as C-SRC, for example, for SARC. So viral oncogenes with have a prefix of V, and cellular oncogenes have a prefix of C. But it was by studying viruses that we first identified that oncogenes existed in the first place. So this is again one of the real strong contributing pieces of evidence for cancer being a genetic disease, because expression of this gene causes cancer. So once we realised that viruses were able to induce cancer by expression of these viral oncogenes, a couple of scientists called Bishop and Varmus in the 70s started to look for cellular genes, human genes, which looked similar in structure and in sequence to the viral genes which were causing disease, to see if we also express these oncogenes which could effectively, when expressed, cause cancer in an individual. And what Bishop and Varmus did is they identified numerous cellular genes which they termed protooncogenes. And it turns out that these protooncogenes are actually involved in key processes that regulate cell growth and cell division. So the reason they're called protooncogenes is that normally, under normal circumstances, these genes and their gene products, their proteins, are doing an important job in the cell. They're not actually causing disease in any way, shape or form. However, they have the propensity to actually go on to cause cancer in cells if mutated. So a proto-oncogene describes a gene such as a growth factor or a growth factor receptor, and we'll talk about the different categories in a moment, that has a potential when mutated to actually drive a cell into a cancerous state, to transform that cell that contains that mutated proto-oncogene. So if you start looking at what types of genes proto-oncogenes are, you will see that, as I've mentioned, they all tend to be involved in key processes around cell division. So many growth factors are termed proto-oncogenes. The subsequent growth factor receptors are also proto-oncogenes. Molecules such as G protein coupled receptors like RAS also function as proto-oncogenes. Transcription factors. Remember, transcription factors are proteins that turn on and turn off gene expression. So these are proteins which can globally switch on a range of different gene expression in the nucleus of a cell. They're also proto-oncogenes, or they can be proto-oncogenes. And key components of the cell cycle machinery, such as the cyclins. Remember, the cyclins are the proteins whose abundance is modulated through the cell cycle and who are required in combination with the cyclin-dependent kinase proteins in order to drive the cell cycle through each of these transitions during cell division. All of these proteins can function as proto-oncogenes, and I've given specific examples of proto-oncogenes that fall into each of these 5 categories in brackets here. What you should notice is a key trend here. If you think about what goes on in a human cell when it decides it needs to divide and undergo cell division, the first thing that needs to happen is a growth factor needs to be sent to that cell to tell it to divide. That growth factor signal needs to be recognised by the cell, and it achieves that using a cell surface receptor. There needs to be an internal cellular signalling mechanism internal of the cell which recognises when a receptor has been bound by a growth factor and then sends a signal through the cytoplasm to the nucleus, which tends to activate a transcription factor in the nucleus in such a way that the transcription factor then turns on specific types of gene expression. In the case of cell growth, gene expression required in order for the cell to enter the cell cycle and start dividing. And finally, obviously, some of the components which are required for that cell cycle progression to occur are the cycling proteins and components of the cell cycle machinery itself. So all of these different genes here are involved in a common pathway which basically underpins the process of cell growth and cell division. So how do proto-oncogenes go from being, if you like, essential components of the cell division machinery to really malignant genes which are causing cancer in the cell, or causing the cell to become transformed? Well, obviously the genes become mutated, but they tend to be mutated via a specific type of mutation which is called a gain-of-function event. So we discussed gain-of-function events during the genetic variation lecture. So this is a mutation which changes the actual function of the target gene product in a subtle way, and in such a way that it leads to that gene product now causing a problem for the cell, which can manifest as a disease. So remember, gain-of-function mutations are different to inactivating mutations, such as what we see occurring in autosomal recessive genetic disorders. So in a gain-of-function mutation, you only need to mutate one copy of the gene in the genome in order to elicit a negative effect for the individual, because remember, the gene product is gaining a function, it's doing something new, something it shouldn't. So the fact it has one working copy doesn't really help it in any way. That's the opposite situation to loss of function mutations, where you lose the ability of one of the two copies to work. However, you still have a backup copy, and in most situations that's sufficient for function to continue as normal and for the person to be asymptomatic. So how do these gain-of-function mutations manifest then? One of the common ways in which they manifest is actually by amplification. So this can be as a result of increased gene copy number. So during DNA replication, if there's an error in the replication machinery and for some reason a region of the DNA is replicated twice, you can get expansion of a gene in such a way that rather having one copy on a chromosome, suddenly you have two. If you have two copies of a gene, that's twice as much of the gene that's going to be produced during gene expression. So you're going to increase the actual amount of protein that's physically in the cell. And you don't have to make a gigantic conceptual leap to see if you have to realise that if you have vastly increased amounts of, say, a transcription factor that's turning on gene expression, then that will result in increased amounts of downstream gene expression activation. And it might be that you end up producing too much of a particular gene, and that then tips the cell into this uncontrolled division of cell cycle. Activation by mutation is possible as well. Point mutations that can lead to gain-of-function events can also occur. Subtly modify the gene product in such a way that it starts working in a way that wouldn't normally work, and that therefore leads to disease. And finally, activation by chromosomal rearrangement. In particular, like we talked about earlier on, if a gene is placed from a transcriptionally silent section of a chromosome, so this will be part of a chromosome in which the chromatin is very densely packaged and gene expression is switched off. If a gene is taken from that particular chromosomal structure and placed via a translocation event in a part of the chromosome which has very loose chromatin structure, so a part of the chromosome where gene expression is very accessible and is occurring at a high rate, that can lead to a gain-of-function event. It can lead to the increased expression of one of these particular genes, and it can lead to the generation of an oncogene from a proto-oncogene, cellular transformation, and cancer. So that's oncogenes. So oncogenes are genes which drive cells towards a cancerous state. So these are the genes which actually lead to the cellular transformation event that we've been talking about, when a cell ceases to be normal and it becomes cancerous. So what are tumour suppressors? Well, in effect, they're the complete opposite. So if oncogenes induce cellular transformation, not surprisingly, tumour suppressor genes suppress it or prevent it. And how were they identified? Well, they were identified by a really nice experiment quite a while ago now, in the 1960s, by a guy called Harris. And what Harris did is he took cancerous cells in tissue culture and he fused those cells with some normal cells from rats, from rodents. I think this picture is actually a mouse, not a rat, but it's similar kind of silhouette, and it's just, let's call it a small rat. But the point is, Harris took some rat cells, which were normal, and he fused them to some cancerous cells. And what he saw, very interestingly, is that the rat cells, which were normal, kind of cured, if you like, inverted commas, the cancerous cells. So the cells which had been transformed in cancerous became normal again. So what Harris inferred from this, what he hypothesised, was that there must be some proteins or something in the normal rat cells which are able to suppress the actual negative proteins, if you like, or whatever's causing the cancerous cell to be cancerous. And it turns out what those proteins are, or were tumour suppressor proteins. Now, if oncogenes and oncoproteins are involved in the business of promoting cell growth, positively regulating cell growth, so that these oncogenes, remember, are growth factors, growth factor receptors, growth factor signalling molecules, etc. It's not surprising that tumour suppressor proteins are proteins which tend to negatively regulate cell growth. So these are the proteins that tell the cells to, whoa, stop, don't divide, wait there, we don't want you to replicate your DNA, etc. Now, you should be aware from the cell cycle that the cell decides during the cell cycle whether or not to divide, whether to enter the cell cycle and commit to a round of division during the G1 phase, and in particular prior to this restriction point in G1. So therefore, it's probably also not a great surprise to you then, if you're aware and if you've been keeping up with your reading on these previous lectures, that some of the major tumour suppressor genes actually turn out to be the same genes which are involved in the regulation of this G1 phase checkpoint. And in particular, two really important tumour suppressor genes are the proteins are PRB and P53. So we've talked at length about both these proteins in previous lectures on the cell cycle, and I just want to now highlight their function as tumour suppressor proteins in addition to their function, which we've already talked about, in regulating cell cycle control in response to DNA damage in the case of P53, and also regulating cell cycle control in response to availability of nutrients, etc. in the case of RB. So the first thing to realise about how tumour suppressors genes interact and fit in, if you like, to the genetic story of cancer is that whereas with oncogenes, the mutations which tend to activate oncogenes are gain of function mutations, dominant mutations if you like, the mutations which are associated with tumour suppressor genes generally are recessive in nature. So what that means is that generally, in order to see a complete loss of function for a tumour suppressor gene, you require both copies of that gene to be mutated in the individual. So in other words, in order for the tumour suppressor gene to completely stop functioning, both copies of the gene need to be mutated and wiped out. And this relates to a famous theory in cancer called Knudson's two-hit theory. And this is a theory that was put forward by this guy Knudson to explain the observation in the clinic for a particular type of ocular cancer, of eye cancer, called retinoblastoma. It's actually it's obviously the cancer which gave this particular gene RB its name. So retinoblastoma tends to exist as a disease, as a cancer of the eye, tends to exist in two different forms. There's a sporadic form of retinoblastoma which contributes about 60% of all cases in the clinic. And in the sporadic form of retinoblastoma, Knudson hypothesized that in order for the individual to start developing the cancers in the eye, both copies of this retinoblastoma gene must be mutated. And this was going to be quite a rare event, and it will probably take a long time for the actual retinoblastoma genes for both copies to acquire mutations, and this explained why individuals with sporadic retinoblastoma didn't develop the disease until quite late in life. Now, in contrast, the other 40% of cases of retinoblastoma cases that present in the clinic are actually what are called familial retinoblastoma cases. So this is a type of cancer that can be tracked through families. And what Knudson hypothesized again was that, well, what if in the familial retinoblastoma people already have one mutated copy of RB? In other words, they've already inherited a mutated copy of the retinoblastoma protein from a parent, and therefore they only actually require a single mutation event instead of 2 independent mutation events in order to wipe out the function of RB and start developing tumours. And if this is the case, if they only need 1 mutation event and not 2, that's a much more common event. It's still very rare, but it's a lot more common than needing 2 independent mutations in the same cell. And that's why familial retinoblastoma presents at a much earlier point in life, and it also presents generally in both eyes, in a bilateral phenotype. So let's think about some of the maths behind this. If you consider that there are around 1 million cells in the eye which are susceptible to this particular type of cancer, and that the mutation rate in those cells, the likelihood of PRB being mutated, is around 10 to the minus 5, and you require 2 independent mutation events in those cells in order for cancer to develop, then you can work out roughly the likelihood of cancer developing in the sporadic retinoblastoma case by multiplying 10 to the 6, which is the number of cells, by 10 to the minus 5, which is the likelihood of a mutation event occurring, and then multiply it by 10 to the minus 5 again, because we require 2 independent mutation events. And if you do that maths either in your head or on a calculator, you will find that the likelihood of that situation arising is around 1 in 10,000. So there's around a 1 in 10,000 likelihood that you may develop retinoblastoma as a disease sporadically. So that explains, 1, why it's quite rare, and 2, why if you do develop it, you tend to only develop it very late in life, because it takes a long time for those kind of probabilities to occur in those cells. Conversely, if you consider the situation where you've already inherited 1 copy of RB, and you ask the question, what's the likelihood of a second independent mutation occurring in those cells, which already possess 1 mutation, then the numbers are much more scary. We've got 1 million cells, 10 to the 6 cells. The likelihood of mutation is 10 to the minus 5. 10 to the 6 multiplied by 10 to the minus 5 gives you 10 to the 1, or 10. So we've got a likelihood much greater than 1. So in other words, it's very likely, in fact it's almost certain, that you will develop these tumours in those particular cells at some point in your life. And because it's such a high chance, relatively, those tumours are probably going to form early in life, and they're probably going to form in both eyes, because all cells of that particular type in either eye have a high likelihood of developing a second mutation and thus going on to develop these tumours. So that's the 2-hit theory for mutation inheritance. That's Knudson's 2-hit theory for the development of cancer, if you like. And it's a general theory that you can apply to nearly all familial cancers, which incidentally generally always involve the mutation of these of a tumour suppressor gene in such a way that it gets deactivated and then allows and facilitates the development of cancer. Another example would be the mutation of the breast cancer susceptibility genes BRCA1 and BRCA2. You can read about that in the core textbooks if you're interested. So that's, if you like, a simplistic way of looking at cancer development. It's a nice model, Knudson's 2-hit theory, and it's important to understand how it relates to the likelihood of developing cancer and can be used to explain the differences between familial and sporadic cancer, both with regards to age of onset and likelihood of actually developing those diseases. But what we actually need to consider when we think about cancer as a disease is a bit of a more complex model than that. And this is what Bert Vogelstein put forward in his so-called Vogel Graham. And this is known as a multi-stage model for cancer. And it basically takes into account these facts. So these are the facts that, in actual fact, in order for cancer to fully develop, and in particular for cancer to become metastatic and go and kill somebody, it needs to acquire several capabilities to get around the multiple defense mechanisms that exist in our body and in our cells. So one of the first things it needs to be able to do is have independence from external growth signals. So if you have a cell that's growing out of control, the first thing the body's going to try and do is to stop that cell from growing, send negative growth factor signals to the cell. So a cancer cell needs to be able to ignore those signals and continue to grow. It needs to be able to evade apoptosis. Remember, P53 is going to want to kill those cells. If the cells are growing in an uncontrolled manner, we can't stop the cell cycle. The next obvious route for the cell is to cause those cells to apoptose, to undergo so-called programmed cell death, because it's better to kill a few cells via apoptosis than allow those cells to continue dividing and for the tumour to take over the body as a whole. So cancer cells need to be able to evade apoptosis. Now, as we'll discuss on the next slide, cells have a finite lifespan, like we as organisms have a finite lifespan. A cell can't divide forever. Eventually it will stop dividing and it will die. So cancer cells need a way of getting around this mortality, if you like. Cancer cells need a way of becoming immortal, of being able to divide forever, if they're going to achieve their goal of continually dividing in an uncontrolled fashion and taking over the entire body. So they need a way of becoming immortal. And finally, if you're interested in growing a very large tumour, one of the biggest challenges that you will face as a cancer is supply of nutrients and supply of oxygen for the cancer cells that are found towards the centre of that tumour mass. So cancer cells need to be able to trigger a process called angiogenesis. And what angiogenesis is the formation of new blood vessels, effectively, in a basic way. So cancer cells within a tumour mass need to be able to promote the formation of new blood vessels. So blood vessels invade the tumour mass and provide the cells towards the centre of the tumour mass with much needed nutrients so they can continue to grow. If angiogenesis doesn't occur, the cells towards the centre of a tumour will eventually die due to lack of nutrients and lack of oxygen. They'll become what's called hypoxic, which means they have a lack of oxygen and they'll effectively just undergo necrosis and die. So the cancer needs to avoid that, and it avoids that by promoting the invasion of blood vessels into the tumour. And finally, if you really want to do somebody in as a cancer cell, you want to gain the ability to become metastatic. So you want to gain the ability to break off from your buddies in the benign tumour, get into the bloodstream, float around the body and maybe lodge in the lung or the kidney or the brain or wherever, and then start forming a new tumour at a distal site. So gaining metastatic potential is another thing that the cancer needs to acquire in order to, if you like, fully complete its journey to the dark side and become a metastatic tumour that's going to kill somebody. So what Vogelstein did is he produced a diagram which produces Vogelstein diagram, which kind of summarises these events occurring during the development of a tumour. And there's a particular tumour in which we understand very well and in a lot of detail the specific types of events that are occurring to enable these different capabilities to manifest in those cancer cells. And that's in a particular type of familial colon cancer called FAP, familial adenopolyposis. So this is a more general, if you like, overview of the Vogelstein diagram, the Vogel Graham for FAP. I just want to talk you through the different events which are leading to eventually the formation of this metastatic malignant cancer here. So the first event in FAP is the loss of heterozygosity for a particular tumour suppressor gene called APC. So this is actually confusingly not the same APC as we talked about during the cell cycle lectures. So this isn't the anaphase promoting complex, which if you recall is a big machine that chews up other proteins. This is a different protein product which is just specifically related to this type of colon cancer. But this APC gene is a tumour suppressor gene, and if you're unfortunate enough to inherit one mutant copy of this APC gene, then much as we saw for RB in the Knudsen 2-hit hypothesis, it's likely that at some point you're going to develop another mutation in APC in the good copy, and this will lead to a loss of heterozygosity, as it's called, for this tumour suppressor, and the beginnings of cancer starting to develop. So it will allow the cells in the colon to start hyperproliferating. At this point, though, what you're getting is a formation of lots of small polyps in the colon. It's not full-blown cancer, it's not dangerous, it's a precursor to cancer, however. In order for the thing to progress to the next stage, we now know that you need an epigenetic event to occur. So there needs to be some kind of event which alters the chromatin structure of the actual DNA in such a way that other oncogenes are activated, which drive the cellular transformation forward and cause the polyps that appear to become larger in size. Further down the line, you get activation via a gain-of-function mutation of the Protooncogenes RAS. Once you activate RAS Protooncogenes, these polyps are now getting quite large. So this would almost be considered abnormal cells, precancerous, and if they were seen by a surgeon, this would raise alarm bells that this individual needed to be monitored very closely for the formation of cancer. Further down still, what happens is we get loss of a second tumour suppressor gene. So we've already lost one tumour suppressor gene, APC. These cells now get hit by a second loss of tumour suppressor gene. So another loss of the tumour suppressor gene, which causes now this precancerous polyp called a severe dysplasia to form in the colon. So this is where, if you like, we're right at the end of the benign tumour. So this is now quite a dangerous point for the patient. This is still benign at this point. The tumour's not going to spread around anywhere, but it's just on the cusp of becoming malignant and metastatic. And all it takes for that transition to occur, that leap from a benign tumour to a metastatic tumour, is the loss of yet another tumour suppressor protein, in this instance the p53 protein. So p53 is a really key tumour suppressor protein. Over half of all cancers have p53 inactivated. That's because p53 is so good at what it does. It's such an important checkpoint control protein, such an important, if you like, overseer of what's going on during the cell cycle. It's often called the guardian of the genome for that reason. Once you lose p53, these polyps cease to become precancerous and become fully malignant adenocarcinomas, tumours, and start to acquire some metastatic potential. You see how the tumour now is invading inward towards a blood vessel here. So I just want to finish by talking a little bit about telomeres in cancer. So this relates to the point I made on the last slide about one of the attributes cancer cells need to acquire is immortality. So how do they actually do that? How do cells which are cancerous get around the fact that normal cells are only able to divide for, say, 50 or so divisions before they effectively stop dividing and die? Well, first we need to understand why normal cells, or adult cells if you like, are limited to cell division in the first place. And there's a massive stack of evidence that suggests that the reason somatic cells, adult cells, are only allowed to undergo a certain number of cell divisions before they actually stop dividing and die is due to the shortening of a special region of the chromosome called the telomere. So all of your chromosomes have telomeric sequence at the very ends, the very tips of the chromosome arms. And what this telomeric sequence is a repeat sequence of nucleotides which is found at the end of all chromosomes in many, many, many copies. So you have numerous copies of this repeat sequence at the end of all your chromosomes. And I'm not going to go into detail now as to what this telomeric sequence does, other than to say that it's important for maintaining the integrity of the ends of chromosomes. And if you don't have sufficiently long telomeres at the end of your chromosomes, the upshot of that is that the genetic information within the chromosome is not stable, mutations can acquire, and effectively the cells will ultimately die. So during normal cell division in adult cells, every round of division tends to shorten the length of the telomere until such a point that the telomeres become so short that they can no longer function properly to protect the end of the chromosomes, and the cells with these extremely short telomeres then will die. So there's an example in a non-disease, if you like, a non-cancerous example of where cells are actually able to divide many more times than we see in adult tissues, and that's during embryogenesis. So during embryogenesis, the actual length of the telomere is not reduced in size. In fact, it's maintained, even increased in some cases. And it's increased and maintained via the action of a protein called telomerase. What telomerase does is it's able to add to and maintain the length of the telomere sequence found at the end of the chromosomes. Now, once we leave embryogenesis and we have adult somatic cells, telomerase activity is switched off in those cells. And that's what causes cells to, if you like, become mortal, to only be able to divide for a certain amount of time. However, interestingly, when we look at cancer cells, what we see in cancer cells is that in many occasions, many instances, telomerase in cancer cells has been switched back on. So it seems as though the cancer cells are gaining immortality, allowing themselves to divide continually by activating telomerase. So you can understand the reason, therefore, why telomerase is a really highly researched area at the moment with regards to trying to understand whether or not we could target telomerase, and by targeting telomerase, prevent the immortality of cancer cells and use it as a direct treatment for disease. It always makes me laugh when I see some of these anti-aging cream adverts. There was one around a year or two ago which claimed that the anti-aging cream had telomerase present in it. I'm not saying it was this particular brand. This is just a generic brand. Other brands are available, obviously, of these anti-aging creams. But it just made me smile to think of people rubbing telomerase into the skin. So this is a compound which has been linked with linked with cancerous cells. So yeah, I think the science advisors for some of these companies need to do the homework a little bit more, really. I'll say that's all allegedly, obviously, just in case anybody from these companies is listening to this YouTube podcast. It's in my opinion. So just to finish then, what we've been talking about today, I've introduced you to a few key terms you need to know the definition of to be able to discuss cancer genetics, such as cellular transformation and benign and malignant neoplasms, etc. And we've talked about the fact that cancer is a genetic disease, and we know that because of the fact that some cancers run very predictably through families. Oncoviruses are able to induce cancer in infected cells via the expression of oncogenes, viral oncogenes, and mutagens which are known to mutate DNA are directly able to also cause cancer. All strong evidence that cancer is a genetic disease. We also have discussed how the mutations which are actually occurring in these cells tend to occur in proteins which are able to disrupt the cell cycle checkpoints. And that these genes which are mutated during cancer come in two flavours generally. Oncogenes are directly able to cause cellular transformation, directly able to lead to cancer. And these oncogenes are generally proteins which are involved and genes which are involved in the regulation of cell growth. Conversely, tumour suppressor genes are generally genes and proteins that are involved in preventing cell growth, or at least restricting cell growth in certain situations. In contrast to oncogenes, tumour suppressor genes generally inherit autosomal recessive mutations, and therefore you need two mutations in order to wipe out the function of a tumour suppressor protein. This relates to Knudson's two-hit hypothesis that we talked about for RB. And finally, telomerase has been linked with the ability of cancer cells to divide indefinitely. And there's research going on at the moment to try and look into in more detail the relationship between the expression of telomerase and the expression of telomerase in cancer cells, and whether affecting that expression might be a possible route for treatment in the future. So I hope that's been useful. As always, look out for the other podcasts on my YouTube channel, Boyne at Bradford. And you should always supplement everything I say in these lectures with information from the core textbooks, and should you wish, wider reading as well. So I hope this proves useful during your revision. Thank you.